Hello, my name is Hannah Kern.
And I'm Monica McDaniel, and today we are going to talk about Photosystem II, the effects of calcium on oxygen evolution rates, and the Tyrosine Z radical signal. Photosystem II is the part of photosynthesis that produces oxygen. For this project, we worked with Dr. Hetty at the University of North Carolina at Greensboro.
This presentation is going to be a brief overview of this poster.
The purpose of this experiment was to determine if calcium is required for oxygen evolution in Photosystem II. We observed the dependence of calcium on oxygen evolution by comparing a sample that contained calcium to a sample that did not contain calcium. We also observed the dependence of calcium by comparing tyrosine radical production by using EPR.
Photosystem II is a key protein complex involved in light energy conservation reactions in photosynthesis. During photosynthesis, PS2 utilizes visible light to extract both protons and electrons in water to produce oxygen. This oxygen that is produced makes up essentially all the oxygen in the Earth's atmosphere. Photosystem II generates two tyrosine radicals, Tyrosine Z and Tyrosine D. The Tyrosine Z radical does not decay when the electron transfer is inhibited in the oxygen evolving complex.
In this experiment, the PS2 samples were prepared from fresh market spinach by extraction with Titrin X100. The samples were prepared with buffers with and without calcium to compare the effects on oxygen evolution in the Tyrosine Z radical. The picture shows an EPR spectrometer. EPR is a technique which depends on an unpaired electron, and this is what we use to observe the Tyrosine Z radical.
This slide shows the dependence of oxygen evolution on calcium. Figure 1 shows the oxygen evolution activation curve, which shows the O2 concentrations as volts versus time. The slope of the graph is used to determine the oxygen evolution rates. Figure 2 shows O2 evolution activity of PS2 is higher in the presence of both calcium and chloride than with chloride only. It is the lowest in the absence of both. Figure 3 also shows the same trend. We also prepared samples in the presence of low pH and high NaCl, which is a method used to remove calcium. The oxygen evolution assay was run in two different buffers, one containing a low pH, high NaCl buffer, and the other using the control buffer containing both calcium and chloride. In the control buffer, oxygen evolution activity was able to be restored. This slide shows the EPR spectra of tyrosine Z. The tyrosine Z radical is induced by illumination and decays during dark adaptation, leaving only the tyrosine D radical. If electron transfer is inhibited, the tyrosine Z signal decays slowly and can be trapped by freezing in liquid nitrogen at 77 degrees Kelvin. The two figures on the left, figures 4 and 5, were samples prepared in the presence of calcium and chloride. The blue curve in figure 4 is the dark adapted sample, which shows only tyrosine D, and the orange curve is the sample after it has been illuminated. Figure 5 shows the difference between the two spectra, which only shows an unknown radical present but no tyrosine in the presence of calcium. The two figures on the right are the same experiment, but with the absence of calcium. And as you can see by the different spectrum, there is some tyrosine radical in addition to some of the unknown radical present. This means that tyrosine Z is formed in the absence of calcium.
This slide shows the Tyrosine Z and Tyrosine D residue signal heights. What these two tables show is that the amount of Tyrosine Z formed is directly related to how much electron inhibition is present at the manganese cluster. In Table 1, the sample with calcium chloride is the same as shown in figures 4 and 5 in the previous slide, and it was seen that no tyrosine Z was noted in this signal. However, all of the other samples that had been treated in some way did show increased amounts of tyrosine Z forming. Similarly, in Table 2, the second set of samples, low pH and high NaCl, were run. These samples showed a similar trend, with some tyrosine Z being shown in both the pH-treated sample as well as the sample where calcium had been removed.
In conclusion, the presence of calcium at the manganese cluster is needed to promote oxygen evolution activity as well as for the electron transfer to tyrosine Z in the Photosystem II process. As well as, these are the references used in conjunction with the research we conducted in Dr. Hattie's lab. Thank you for listening.